Hello, my dear lovely students. I decided to explain the all biology 6 book for you this year. Sometimes it is hard to make a review for you, and you have maybe different questions. So to help you and to give opportunity to you, anytime you need to review the lesson, you can watch these videos. And all this biology 6 book, I'm going to explain it word by word for you in videos which made up of only about 10 to 20 minutes. So tonight bore you a lot of videos will be shorter. In all biology 6 book, there are 5 chapters we're going to study or are responsible for ministry exam. Chapter 1, the cell. Chapter 2, tissues. Chapter 3, reproduction. Chapter 4, embryology, and Chapter 5, genetics. Let's begin from Chapter 1, the cell. In this chapter, we're going to study introduction. There are some historical information we're going to study on about scientists and their contribution to the biology. And then after the cell theory, 3 main stages of cell theory that we're going to study. And then cell size, we're going to study how much cell is big or small, is enough to be seen by naked eye, or some examples which we need microscope to observe. And then eukaryotic cells and prokaryotic cell. We first focus on prokaryotic cells, give example, a study on structure, but it is too short about eukaryotic cell. Each organelle of cell we're going to study with the focus on their location, structure, function. And any structure in this book, if you know their location, structure, and function, you can give answer to any question about it. And by cell activities, we're going to study what happens in cell, how these organelles perform their activities. In cell division, we're going to finish the chapter, and it consists of amitosis, mitosis, and meiosis. Let's begin. Introduction. As I say, we're going to study or read each word one by one, and we'll underline, ask questions, make comparisons, draw pictures for you as we study during the lesson. The cell is considered as the basic unit of all organisms. Generally, there are 2 types of cell from structural point of view: the prokaryotic cell, as bacteria cell which lack nuclear membrane, membranous organelles. The second type is eukaryotic cell, which is larger than prokaryotic cell and has clear nucleus surrounded by nuclear membrane and cell organelles. In this paragraph, what we're going to know, there are 2 types of cell according to their structure: prokaryotic cell and eukaryotic cell. There are also some differences between them. They summarize it in this paragraph. Like what? Like prokaryotic cells they lack a nuclear membrane, while eukaryotic cell has a clear nucleus by a nuclear membrane. And another difference, they have no membranous organelles, but eukaryotic cell, they also have cell organelles. As another example, we can give bacteria cell as example for prokaryotic cell, but for eukaryotic cell, we can give example as human body cell, or animal cell, or plant cell. The cell has been the center of interest for a long time. Scientists have a major role in development of cytology. Development in cell study can be briefed as follows. As historical introduction will focus on name of scientists and what they do. The cell was not known when the German scientist Anton von Leeuwenhoek invented the microscope, because cell was too small to be seen by naked eye, and there were no any equipment to observe the small objects. So there were no information, even no name of cell until that time. But Anton von Leeuwenhoek invented the microscope. Maybe may or could be the first to see the cell, but he didn't use the term of cell or didn't identify or describe it. But after one Leeuwenhoek, Robert Hooke used same microscope, did same observations, looked and studied on structure of oak tree cortex, take a cross section, observe it under the microscope, and he saw something look like beehive. We know beehive consists of these hexagonal structures, and because microscope was so simple and cannot see many details in cell, but plant cell covered by a thick cell wall, and clearly borders can be seen by similar microscopes too. So he divided these empty units as cell. So the first scientist who used the term of cell or founder of cell is Robert Hooke, but not one Leeuwenhoek. And about 100 and more years after, Robert Brown, because as time passes, new microscopes are invented, so can see more details about cell, and he see some dark spherical structures in center of cell and call them as nucleus. So for Robert Brown, we need this name, and we need to know he discovered the nucleus and described it. Matthias Schleiden and Theodor Schwann, two scientists, as you know, one of them studied on plants, but not only one plant, like Robert Hooke, many plants are observed, and at the end of this observation, concluded that all plants consist of cells. And Theodor Schwann studied on animals, different kind of animals, take different sections from them, observe it under the microscope, and at the end also he concluded that all animals are made up of cells too. So studies and researches into cytology have increased. Means research, observation about cell increased. How? By advancing in microscope, light microscope, and then electron microscope invented. Light microscope can observe, magnify object about 100 or some thousand times. By electron microscope, 100,000 times or million times can show us it bigger. And as new better microscopes are invented, cell could be observed in higher details, more information unveiled, and it becomes a branch of biology, and we call it cytology. So we can define cytology as the branch of biology which study on cell. This Matthias Schleiden and Theodor Schwann together, they set the cell theory. Cell theory, some informations which consist of three states, we can see A, B, C, and it's set by Matthias Schleiden and Theodor Schwann. These two scientists studied together, and they said, if we remember, Schleiden said all plants, and Schwann said all animals are made up of cells, and together they say all organisms are made up of cell or cells. Cell because some of them unicellular, cells about organisms which made up of many cells as multicellular. And B cells are the basic structural and functional unit of all organisms. We can use this B as a definition of cell. So anytime in ministry exam ask about the cell, we not use this first sentence as a definition of cell, but we use the second state of cell theory. Cells are the basic structural because all organisms are made up of cells, and functional also because all organism body activities depend on reactions happen in cell for all organisms. In C, one cell comes from another cell by means of cell division means when any cell cannot be produced and manufactured in laboratory or in a factory, but only one cell comes from another cell. But how? By cell division. That's what we're going to study at the end of chapter 1. This 3 stages, the main stages of cell theory, also summary of chapter 1. What kind of question can be asked? We need to know the name of Matthias Schleiden and Theodor Schwann, because it can be asked as fill in the blanks in ministry exam questions. Also can ask you about the 3 stages of cell theory. They are important. And option B, we need to know its definition for cell. I said the definition for cell, we have to use that one, because as others is used as the smallest unit of life, building blocks of living things. They are all true, but so simple definitions for cell. We need to use definitions in our book for this year. So anything out of the 6th class book is not true, but anything you write from this book can be answered the question in ministry exam. Cell size. Cells vary in size. For example, diameter of frog is 1 millimeter. It can be seen by naked eyes. However, it can be seen by naked eyes, but cannot be studied or observed in details by naked eye. But most of cells are smaller than this, like human egg also smaller. We need a microscope to see it. But human cell about 100 micrometer, like on the edge of being seen or not seen by naked eye, or it is better to observe it by a microscope. Cells have specific specialties to increase efficiency of various functions. That means according to their function, there are different specialties for cell. As what? Having ability of contraction, relaxation in muscle cell, or having ability to transport oxygen in blood cell, or having ability to transport impulses in nerve cells. We need microscope to see cell and microorganisms. Microorganisms means that cell can be seen by light microscope, but inner component of cell like organelles, viruses, organic particles can only be seen by electron microscope. From this picture, we see this object in picture divided into 3 groups. Some we can be seen only by naked eye, or by light microscope, or by electron microscope. What is important for us to know at least one example for each of this group, but it is better to know more than one, maybe 2. And it is important to use the example found in this book, but not any example you know. Prokaryotic cells. It is undeveloped cell. We said there are 2 types of cell in a structural point of view. The first one, prokaryotic cell, it is undeveloped, primitive in structure, means simple, characterized by the followings. Prokaryotic cell has a genetic material without a membrane and is called nucleus zone. And if we draw simply a bacterial cell, a rod-shaped bacteria, genetic material, all organisms, they have some amount of genetic material, but without a membrane. There is no membrane which cover the genetic material in cytoplasm, and genetic material is free. So the part of cytoplasm with genetic material included, it can be called as nucleoid, but not nucleus, or nucleus zone. That means zone of genetic material. Prokaryotic cell cytoplasm has no membranous organelle like Golgi bodies and mitochondria yet, but there are only ribosome. It has ribosome in form of small grains in this form. even ribosome we're going to study also in eukaryotic cells. This ribosomes, even they are smaller than ribosome in eukaryotic cells, and they build proteins. Means, or we said ribosome, it's producer of proteins in prokaryotic and also eukaryotic cells. See, prokaryotic cells is represented by blue-green algae, bacteria, and mycoplasma. These three examples, they are organisms which cell structure is prokaryotic, means their genetic material in their cytoplasm is free and not covered by a membrane, or they have no membrane covered organelles. And they all belongs to kingdom Monera. What is important for us, we need to know each of these organisms. They are example for prokaryotic cell, also they are example for kingdom Monera, or kingdom Monera by itself also example for prokaryotic type of cell. Each bacterial cell represents a prokaryotic organism. It is surrounded by a solid wall consisting of chemical compounds, protein, lipids, polysaccharides. These chemical substances are important. Why? Because in next part when we study on cell wall in plants, in a comparison, cell wall made up of different substances, but cell wall in prokaryotics are different chemical substances. So we need this information. Behind this wall there is a plasma membrane. It's a fluid membrane surrounding the cytoplasm, which contains the nucleoid. As we said, nucleoid is genetic material which is free in cytoplasm of prokaryotic cell. There are no nuclei or nuclear envelope, unlike eukaryotic cells. Means one main difference between prokaryotic and eukaryotic cells, in prokaryotic cell having no nuclear membrane. Cytoplasm also contains ribosome, also we said because it's built proteins, what is producer of proteins. Some type of moving bacteria, they may have flagella. Flagella, we can see this long filaments which connected to prokaryotic or eukaryotic cell, but generally it used in movement. In addition, in picture we can see another structure like sex pili. We will study it in chapter 3. Bacteria use it in reproduction. And mesosome, its center of producing energy, represent the mitochondria in eukaryotic cell. It consists of some folded membranes. A part of plasma membrane has enzymes and produce energy for bacteria cell. Cytoplasm is jelly-like fluid which fill the cavity of prokaryotic cell. Nucleoid, we said nucleoid, genetic material which is not covered by membrane. Plasma membrane, cell wall consists of proteins, lipids, and polysaccharides. Flagella for movement, fimbria or cilia, same structure, tiny numerous structure which connected to the wall of bacteria and use it for movement. This table summarizes the structure of prokaryotic cell, but when question asks us to explain the structure of prokaryotic cell, we need to know information always study about prokaryotic cell, but not this table. Cell covering, but we can use table just to remember. What cover the prokaryotic cell? Cell wall and cell membrane. In cytoplasm, what found in cytoplasm of prokaryotic cell? A genetic material without membrane and some non-membranous organelle ribosome to produce proteins. So what added to prokaryotic cell? Flagella in some bacteria for movement, fimbria or cilia also used it in movement, and sex pili which did use it in reproduction that we're going to study in chapter 3.